So let's see, what's on the agenda for today? So first of all, next week, special guest Nascent Novice is going to come, and we're going to talk about uh, something a little bit different. We're going to talk about vampires. So moving into the literary space, I think vampires are technically, pretty fantasy, but fantasy and sci-fi, I feel like there's a bit of overlap. Who doesn't like vampires? And vampires have been around in pop culture for a very long time. I feel like we're maybe not in a vampire moment anymore, but we're just coming off of one. So close enough. Just gotta move some things around. There we go. That should be an interesting discussion. Yeah, it should be. Yeah. Basically, he is very much on top of the literature, and I am very much not. But it's got me reading about some cool things, drinking blood and what that would mean, the sort of disease of vampirism. That's kind of fun. But yeah. All right. But what I actually want to talk about today, for the most part, is just Twilight. I don't know if we'll be talking about Twilight. We'll see. I don't actually know. Um, Although Twilight does have one thing that I feel like no other vampire properties have, which is that shiny skin sort of thing. That could be interesting to talk about. Um, anyways, so potentially the last stream on gene editing, DNA editing, that sort of thing, because I promised Managard that we were finally going to talk about Stargate. We got so distracted. It was on my list of like number 2 thing I was going to bring up, and then we never got to it. Like either of the past 2 Wednesdays, but that's okay. Um, I love Stargate's. Well, I love Stargate, period. But I love Stargate's use of DNA because that was out in a really fun time in just society where it's like, okay, we can kind of like sequence DNA. I feel like actually, I guess I can look it up. The episode where this particular device showed up, it might have come out. I wonder when it came out, before or after the full human genome was sequenced. Let's see. So Sg1, yeah, so this was in Stargate Sg1. So background for this device, it's a device built by a species of human that are long, long gone, but has been co-opted by the non-human antagonists of the early 2/3 of Stargate. So where would you start watching Stargate? You never did so because it just never happened. That's a good question. So you could start with the movie, actually. There is a movie. So Stargate Kurt Russell. So Stargate with Kurt Russell. This movie. Oh, Brian, you have to watch it. Yeah, Stargate with Kurt Russell. Have you seen, David, have you seen, what's it called? Independence Day? Independence Day was supposed to be the sequel to Stargate, this movie. It was just rewritten and recast. So it ended up not being the sequel. But I would start with the movie Stargate and then just start from episode 1. There's some iffy stuff in the first couple. Oh, you haven't seen it? That's fine. You don't need to. Just a little bit of movie trivia. There's a couple iffy things in the earlier bit of Stargate. Okay, so Benjamin is suggesting, also future guest, is suggesting movie, maybe in all caps because it's required, then a curated Sg1 list, all of Atlantis, and then universe. Take a copy of Origins and then throw that copy in the garbage. True. I couldn't actually watch too much of Origins. And then, useful information. David, I hope you're writing this all down. You're getting everything you need to know. SG1 is currently on Netflix. Atlantis is on Hulu, and Universe is on demand on Pluto. Poor Universe. Yeah, curated list of SG1 is kind of, yeah, I see where you're coming from. There are some episodes that don't age too well in the early seasons of SG1. So I would say if you start not liking that particular episode, just skip it. But, past season like 2 or 3, I would watch all of them until you get to like 9 and 10. And then you can decide whether or not you want to jump ship. You've been more of a Matrix, Terminator, Star Trek guy, all the stuff that's firmly rooted in modern day society. SG1 is great. I think there's no other sci-fi that has been as popular in recent memory, where they get the everything kind of gets away from them eventually, but for a while they do a really good job of being like, okay, this is actually a secret thing, no one knows about it, it's going on in our real universe, until they start shooting spaceships out of the sky and stuff like that. Then it's kind of hard to say like, oh, how does no one know about this? You can go back to the blah episodes of SG1 later. That's fair. IMDb ratings is pretty good for that. Yeah, that's probably right. You also love the works of Philip K. Dick. I've only really... did I read part of Man in the High Castle? Maybe, but I've watched all of Man in the High Castle. That was Philip K. Dick, right? Listen, if you've stuck it out on my stream for these past couple weeks and enjoyed the stuff that we've been talking about, when I actually do get to talking about some sci-fi, just believe me, Stargate is probably for you. And yeah, Atlantis is perfect, more or less. Universe is different, but also super good. Philip K. Dick. Why did I look him up? I know he wrote a bunch of things, but is Man in the High Castle. Yeah. I need some water already. Water or beer? I've got to be good tonight because tomorrow's my first day of class. In all honesty, maybe I should be working on that instead of streaming, but it's okay. It's syllabus day, so I'll be able to go in, go over what I have on the syllabus, because that's not even necessarily finished. And it'll be a fun time with all 5 or 6 of my students in my very small upper level course. You'll see if you can catch some Stargate. Thanks. Yeah, it's sort of linear. You kind of have to do Stargate SG1 until you get far enough to start Atlantis, and then at some point you can dive into Universe. Yes, I have chosen beer. I've chosen a sour from Sierra Nevada. This is pretty good. I'm going to have to hydrate with actual water, though, eventually. All right. What was I doing? Okay. So the original air date of this, it was in season 6? I guess it was in season 6. For some reason, I thought this was an earlier episode. So one of the antagonists, Nirti, attempted to create a higher evolved human, for whatever that means. That could be a whole discussion in and of itself. And she attempts to do this using an ancient machine that alters DNA. And I'm just surprised it's in the 6th season. My memory is not that good. So January 15th, 2003, I think the Human Genome Project completed in either 2000 or 2001. Oh, never mind. I thought it was 2001. Okay, so they were pretty contemporary. That's kind of cool. Let's see. Where? Completed? That's the T to T project. Oh. Come on. Completed in 2003. Well, if this was released January 15th, 2003, I doubt that the writers and actors wrote it, then acted it, and then produced it in only 2 weeks. So I'm sure this was in production before it was actually released, but people knew about it. Get some German beer soon. It's just the best. Totally agree. 100% agree. Did you say you were German? I spent like a semester in high school in Germany, and I was the age to drink beer at that age. Like when I was in Germany, I was still pretty shy about it, but my classmates were not. That was a lot of fun. And yeah, German beer is really good. Tastes totally different than the German style beers we have in the U.S. Ah, du lebst in Deutschland. I see. Or, excuse me, entschuldigen Sie mir, Sie leben in Deutschland. We don't know each other. And that's the most German I've spoken in like 6 years. Okay. She's behind. Oh, Nirti. Nirti is behind other earlier episodes as well. Yeah. She's always up to her thing, trying to make a perfect human, which is super, like in hindsight, wow, she would have been canceled so fast. Uh, I guess we don't really want to go into like what does that even mean? A perfect human? I don't know. Good pronunciation. Oh, thank you very much. Oh, hello, Miss Frizzle. Welcome. I was just saying how I should probably actually be working on my class because my class starts tomorrow. But here I am, talking about Stargate, drinking beer, having fun. Okay. So, as long as this isn't flipped, one of my favorite things when I first came to this is that the DNA is actually going the correct way around. So DNA has a handedness. It's either left-handed or right-handed. Right-handed is correct. Left is wrong. This is right-handed. I just really wish I had tried to find a higher resolution of this particular screenshot or this particular scene. But I just can't because I really want to know what they're doing. So this is the thing about, just any... There's a couple things that Stargate does fun and good and then does kind of bad, and this is one of them. So this represents a very small portion of the genome. Oh, have a great day. Thank you. I will have a great first day. I'm looking forward to it. So let's see, what is this? 1 2 3 4 5 6. Okay, 6, and then 1 6 12 18. So it looks like they're showing 30 bases right here, and ostensibly what Nirti was doing in this episode was going through and making little modifications here and there. I wonder if there's actually a YouTube clip that I can find. Which, by the way, Manogard, thank you for the Spock clip. I haven't had time to actually make it integrate it, so that doesn't quite work yet, but it's saved to my computer. I'll do it. Nirti Stargate DNA resequencer. Not sure why this would be. Maybe. A protocol for DNA sequence analysis. Okay, let's make sure this is muted. Theater mode. You just checked the episode. Oh, and you don't think the image is flipped. It looked the same. No, this is just a little clip show of her. Here we go. Wait. So here she goes and activates it. Yeah, that's right. They have it right. They who was I talking to? I think I was talking to someone via chat. No. My memory is just so bad. I was talking with someone. Oh, this was on Discord. Yeah, I was just talking with microbloganism on Discord about who also is a researcher or scientist of some kind. And we were talking about how it seems like when you just search DNA, in fact, I'll do that, DNA, and we look at images, it often, but not always is the case that they, like within this, it'll always be like some incorrect ones. So, although now that I say this, all of these are correct. So, of course, it's proving me wrong. This one's incorrect, that one's incorrect, that one's incorrect. So I had to scroll down a little bit, but it seemed like there was, this one isn't even close to being correct. There was like a time where, for some reason, all the DNA everywhere in like art was just backwards and wrong. So that's why I'm surprised that they got it right. All right, go back to this. Wait, I really wish I could see. No, come back. I don't know. Okay. I was really hoping that they actually grabbed like a DNA structure or something like that. And we're just using that as the 3D asset. Well, this could be, let's see, pure as gold. You'd rather have 2 rings of gold. So I think this is a purine. So actually, I guess they could have actually got it. Yeah, we don't want to see the actors, just the DNA. Exactly. They should release the actorless cut of this. Oh, wait, this is Nirti's, the actress who played Nirti's personal channel. What's on her YouTube? Okay. Go follow her, I guess. Yeah. Oh, okay. Okay. Maybe they did grab some. They're just not quite right. Oh, and still, again, I want to zoom into this, but I just don't. Yeah. So, like, what is she? Okay. Okay. So, in this scene, she presses some buttons and apparently zaps even fewer DNA nucleotides. Wait. Oh, like 3 right there? And that's it, I guess. So the thing about this is, uh, I like the concept of a DNA resequencer, which is a great sci-fi term. In fact, I'm going to search it, resequencer, just to see what pops up. Oh, it's just Stargate. There was another one. Yeah, there was another one in Atlantis. So it comes up multiple times. This is just the same place I was before. I love the idea of that. And anyways, what I was talking about before is this came right at when we finally sequenced the human genome. So Stargate was just on that line of like, okay, the possibilities, the door is literally opening to all of this crazy genetic manipulation, that sort of thing. It was kind of in the cultural zeitgeist at the time, or at least it began. I feel like now, if you throw in a like DNA based subplot to somewhere, to some like, thing, you wouldn't be too surprised. It doesn't feel out of place. Yet, when you think about just a couple years earlier in another super popular sci-fi show, The X-Files, I remember the time that they dealt with like DNA editing and resequencing. They basically made like a spoof of Frankenstein, like the original or one of the original movies, the black and white one with the burning windmill. They made fun of that, except that they were talking about Drosophila researchers and Hox genes and all these things. And it went from like outdated to all of a sudden in Stargate, like, this is pretty well dated. okay, they get a couple things wrong. Like you probably can't just change a couple bases and then have it affect the person immediately. Which they do say that people have to go in for multiple sessions, and Nirti is they get it right where she's like tinkering with people's genomes, and she doesn't ever seem to get it quite right, and she always has to go back and change things ever so slightly more and more and more, uh, which is great because we don't know, even if we had this machine, we don't know anything close to what we would need to do anything broadly useful for humanity. Like we could maybe cure a couple diseases, but that's about it. Like I just wonder what the current state of technology is. So if you go to PubMed, what would this be? Whole organism gene editing. Because right now, if you want to edit someone's genes or some things' genes, you can't just stick them in a flashing light machine and do it all at once. The problem is that even if we do use the gene editing technology we have now, like on me, it's only going to change a small proportion of my cells. Ooh, large-scale multiplex mosaic CRISPR perturbation in the whole organism. I don't know if that's what I'm looking for. Systemic genome editing of the genes on zebrafish chromosome 1 by CRISPR-Cas9. Let's open that one up too. It's from 2 years ago. This one's newer. So they report an inducible mosaic animal. So inducible mosaic animal for perturbation. So a mosaic animal is an animal, like a research animal, who has had mutations induced, not everywhere. So that's why they're a mosaic. It's exactly what it sounds like, except the mosaicism is in their cells. Ah, Brian, you've sent me something. Somatic cell genome editing. So the NIH has a common fund program working to improve efficacy and specificity of gene editing approaches to reduce the burden of common and rare diseases caused by genetic changes, right? So they are trying to find effective and safe genome editing tools in non-reproductive cells of the body. Yeah, so we sort of talked about this, I think, like 2 weeks ago. Like there's a difference between changing the cells of the body and cells of the gametes. So if you change the cells of the gametes, which are cells that become sperm or egg, you're going to pass the changes on to offspring. If it's in just the body, the somatic cells, it's just that particular organism. And I feel like that's generally more safe because any side effects that you didn't know about, or even getting into like the idea of consent to DNA editing, kind of go away if it starts and stops within one person. Cool. So they're actually doing a consortium. Who do we got here? This is the worst ever key, especially since the University of Wisconsin-Madison is about 3 hours north of where it actually is. That's silly. There's a video down below, but you haven't watched it yet to see if it's useful. My music's too loud. Hold on. I'll try the video though. Ooh, that was gonna be loud. Hold on.
Diseases are caused by changes or mutations.
Can people hear that? Is the right channel unlocked? Probably not, actually. That might have been really loud.
To the body's
Yeah, probably not. Okay, now you should be able to hear it.
DNA. What if we could treat all these diseases by diving into our living cells to correct the mistakes?
Ooh, they're just going to go by that? Look at that. What do we have here? We have the nucleus. We have some endomembranes. We have some like actin filaments or something like that. Actin? Yeah, actin. Is that a ribosome? I think that's a free-floating ribosome. Oh, mitochondria. Little blobby vesicles.
The Somatic cell genome editing
They got their DNA the right-handedness.
program aims
Although that's a really... I don't like this just scissors. Oh, it's a person. But it looks like they're cutting the person.
to make that happen. Recently, researchers have made great
The DNA is the right way?
progress in correcting DNA mutations using a technique called genome editing, and the first tests of genome editing for human diseases are starting. However, there are still some challenges to achieve safe and effective genome editing in patient cells. For example, the DNA in millions of cells needs to be corrected to treat many diseases, and getting the editing tool
So what they mean is what I said, like you have to correct enough of the cells to make it worth it.
into different types of cells, such as those of different organs, will require tailored delivery techniques and packages.
So we have, I think, just 3 viral vectors. And then there's also, I don't really understand this technology, but you can basically shoot gold particles, which are non-reactive, into patient tissue or into tissue. And these can be coated in DNA or something like that.
The delivery techniques must be specific to enter the right cells and avoid the wrong ones, such as the cells of other organs or reproductive cells like eggs and sperm. Additionally, different types of editing tools may be needed to correct different types of mutations. The Somatic cell genome editing program.
Oh, is this also supposed to be an S? Oh, it is. The red is the S. It's an S, it's a person, and it's a DNA.
Or SCGE, was launched by the NIH Common Fund to develop quality tools to perform safe and effective genome editing in human patients. The SCGE program will make more genome editing tools available to researchers, develop better package.
Wait, so what were they showing? So, I mean, they don't really... Nanotubes and nanoparticles can enter any cell easily. Maybe. Please bring the scheme in India. Please bring the scheme in India. Is there, and there's a hidden comment. Oh well.
Tools of
Yeah, I don't really know what these 3 are supposed to be. I don't remember what Cas9 looks like. Protein data bank. Cas9. I didn't see an RNA, so that's why I'm wondering. So that's Cas9. But they have it as like a blobby sort of thing. Cartoon. I want surface. Or not? How do I add a surface? Come on. Oh, there it is. So, oh, maybe that is it. Just kind of scoot that. Oh. Hard to tell. I give up.
available to researchers, develop better packages to deliver the tools to the right cells, design new tests for the safety and efficacy of genome editing, and make all of the information available to the scientific community to drive future discoveries and cures for patients.
All right, so I mean, I'm not surprised that the NIH wants to promote that. Okay, but have we actually done something like this yet? It seems like this group from China, the Gene Editing Center, might have done something. So they have an inducible mosaic animal for perturbation, a transgenic platform enabling in situ CRISPR targeting of at least 100 genes in parallel throughout the mouse body. Okay, it combines Krelox P and CRISPR-Cas9, germline transmitted transgene. Yep. Oh, interesting. Okay. So what they have is not necessarily oh, Brian's sending me stuff. Oh, I think I've read this before. I think they read that for a class that I led. I'll go back to that. All right, so this isn't quite the same. They're doing something where they're changing a gene in one gene in every cell out of 100 genes that they've targeted. So if they have 100 cells, they expect each cell to have one different gene targeted. Okay, so that's a little bit different. Still cool. So what's this one? A whole genome knockout for any species has rarely been achieved. They performed systematic knockout of 1,000 genes on chromosome 1 of zebrafish, successfully mutated just under the number. Okay. They just did it. Okay. That's not interesting. That's something else. Okay, Brian, but you might have sent me something more useful. So many metabolic disease models still depend on laborious germline targeting and are further complicated by the need to avoid developmental phenotypes. They generated somatic mutations in the adult liver using CRISPR-Cas9. They targeted a particular protein, which when deleted leads to severe hypercholesterolemia, cholesterolemia, and atherosclerosis, a word I've never been able to say. Let's see. Okay. So wait, what did they do? I'm just going to skip to the end, see if they have a... I see. See if they have a cartoon of what they did at the end. Probably not. I don't know if I want to read this whole thing. I feel like I remember this, though. So here's the discussion. So they use an adenovirus CRISPR to disrupt a particular gene, and then they disrupt another gene. I see. So okay, people they're doing the somatic genome editing. That's really just what I wanted to find out: is are we doing this? It's going to be hard to. Yeah, I mean, people are doing stuff. Ooh. Ethics of creating and using human-animal chimeras. Whole organism genome editing, targeting large DNA insertion via something weird. 2015. A 3-person paper that I haven't heard of. I doubt it's that exciting. Anyway, suffice to say, we don't have a a fancy light show machine that changes DNA. We're still using molecular tools. I don't know. This is a part of physics that I don't know enough about. So we have optical, what are they called? Optical tweezers. Yeah, where we can use a focused beam of light, or maybe not light, but a laser beam, in order to manipulate small molecules. These images are all horrible, but I guess this is effectively what it is. Give me a bigger picture, please. And I forget how old this technique is. 1988? So this is a real thing, right? It's just... This machine probably is not pumping the character full of enzymes and proteins and DNA to like act as a template to be changed out with the DNA that you're altering. So I feel like the disconnect between like this type of like super technology and what we can do today is that all we can do today is manipulate the genome and manipulate the cells, usually with stuff that's like biological or chemical in nature. So there are chemical inducers of mutation, but they're not targeted. Let's see, what's it called? Is it called EMS? EMS mutagenesis? Is that what it's called? Yeah. Ethyl methane sulfonate. So, yeah, EMS. So you add EMS to cells, and effectively what it does is, it modifies certain bases in the DNA so that when they are then replicated in just the course of making one cell into two cells, a mistake will be made. So the EMS will, for example, bind to the guanine or a G, and it will cause it to misrecognize a thymine. So you go from a G to a T. And I guess it does the opposite too. It attacks a thymine, a T, and can go to an A, or a G, I mean. You just finished an EMS screen? Really? Like, did you do the EMS yourself, or is it something that already existed? And also, did you find anything? Because, see, I know that people do still do EMS screens because it's really useful. So an EMS screen, for those who don't know, is you basically add EMS. It's a mutagen. It causes mutagenesis. It's just random. You do EMS by exposing organisms to EMS. You let them breed or whatever and do keep single lines going. At least this is in the organism I work in. So that you do it in such a way that on average, there's only like one or two mutations every like certain number of bases, I think it is, or even maybe only one major one per like genome. I forget exactly what it is. Maybe it's one mutation per gene. I don't know. Anyways, and then the screening part is where you are like, okay, I want a particular phenotype, and you look through your big batch of mutated organisms and look for that phenotype. And that can potentially tell you something about what you're actually working on. And no, you didn't find what you were looking for, but it's easy in C. elegans. That's too bad. Okay. So, anyways, I was talking about optical tweezers because I feel like the only way that you'd actually be able to manipulate a genome on like the time scale that they do, or like with the like precision that they do it with everywhere, is I feel like you have to physically go in and somehow go in to each cell, find the exact places that you want to change and change them directly. Like directly, not waiting for an enzyme to bump into it and hopefully do something that you're hoping to look at, or alternatively just splash a bunch of mutagenic chemical onto it and hope that something cool happens, because that is not at all what Near 2 is doing. So there was another one. So there was Atlantis too. 12 strand. What? Okay. This is utterly unrelated, but I do have to click on this link now. No. In this meditation, chelantic frequencies will help activate your 12-strand DNA code. What? What? This person is a student who seriously studies chelantic science. What? Hold on, I'm actively going down a rabbit hole. I detect no 12-strand DNA activation from this music. Oh, okay. What? I'll unmute it in a second. It's just that. It's just an hour and 11 minutes of those sounds. What is key? Oh. Oh, is that what you just sent me, Brian? This book? Oh no. If I skip, we'll only activate 8 to 10 of our strands. What? Hold on. I mean, usually, I don't mind engaging with this sort of stuff, because I find it extremely interesting what people think other people want to. Okay. I'm gonna read part of this. The picture in the book that shows the violet sphere that I am holding in my hand contains the frequencies and the keys that will become the Merkaba crystal spaceship to carry the cosmic twins back to Tara, Gaia, and beyond. The violet sphere was created by following the directions of the Elohim angels and Syrian council. The picture was taken during the moment of the stellar wave infusion of the violet wave from the sun Achelon, and stellar wave activation of the Syrian spiral in 2008. Oh, okay. The stellar waves activated our 5 DNA and began the initiation of the 12 subharmonics into 6 DNA. What? I just want to know where this number comes from. Oh, it just keeps going. What are people saying? Laughing. Oh, no. What other link is this? Ah, okay. Barnett, I think that was the author. She's the author. I can't take that away from her. Learned that she was Mary Magdalene in the flesh when she was given the assignment of opening the Pacific Ocean Ascension portals. Okay, cool. Very cool. No, not author in quotes. Author. I mean, she did have to write this. Cosmic Mystery School of the Omniverse Publications. Ooh, crystalmagicorchestra.com. This has nothing to do with Stargate. crystalmagicorchestra.com. Oh, it's wonderful. Hold on. I love when people misuse DNA. Oh, okay. DNA activation. She might explain. Oh my gosh. Okay, that was already... Huh. This is one of your favorite rabbit holes so far. Well, great. Good, I guess. Maybe bad. Okay, I'm going down a sub-rabbit hole for a second, so you have to remind me when I start climbing back out of it. So I have not yet received the kit or sent it back, but I did purchase whole genome sequencing for myself. Okay, and I was thinking, okay, what could I do with that information that could be fun? And I was thinking, like, what could I do that's artistic or something like that? So then I did some calculations and tried to figure out, like, could I print out my DNA, like, on a piece of paper? Or what I knew was going to be many, many, many pieces of paper. If I were to print out 11 by 8.5 sheets where a single dot, barely visible by the human eye. Hello, science streams. Welcome. You caught me at a very awkward time in that we're talking about some non-science. I'll put this up if you want to skim over it while I'm talking about something else. Yes, Alt Miss Frizzle, I did do it. I purchased whole genome sequencing for myself. Anyways, like, could I actually go print out, like, my DNA? That book would be like 3 times the size or length of the entire Encyclopedia Britannica. So that's probably not going to work. And then I even thought, like, okay, well, 99% of it is probably the same as everyone else, right? Because that's how it works. Even just doing the differences is going to be quite a lot. Now, here's my point. What if I did actually print it out as just letters, right? Letters. And what if I printed it out onto a digital book? Could I publish that digital book on Amazon and sell it to people? Like, could I recoup my $200 if I sold, like, my entire genome for $10, right? $10, you can have complete access to my genome, start to stop, with the caveat that, telomeres, centromeres, pericentromeres, probably not there. I don't know if that's a good or bad idea to try. Probably a bad idea. But I can't think of what specific reason. Like, oh, the government could have your DNA. Well, government probably has my DNA. Or, like, you'll know something about my health. Well, it's like, well, not really. I mean, as far as I know, there's nothing. I don't have any genetic diseases that I'm aware of, because I've already done a little bit of genetic ancestry DNA testing on myself. And, I have the occasional, like, oh, increased chance for this cancer, decreased chance for this other cancer. I have red hair. I have pale skin. Like, not really that much to take from that. So, I don't know. Actually, can we do a poll? I want a poll of whether or not I should sell my DNA as a book. Because if people are paying $28 for the 12 DNA Music Spheres Ascension program from the Cosmic Twins Diary with Keys to the Universe by Dr. Angela Barnett, certainly I could sell... Wait, comma. Oh, for some reason I'm on, like, a non-English version. Certainly I could sell, like, my entire DNA from Dr. N for $10 or something like that. Oh, thank you. Can I vote? I'm voting. My vote's yes. What exactly is her doctorate in? Yeah. Doctorate. Let's see. Well, she learned that she was Mary Magdalene in the flesh when she was given assignment of opening the Pacific Ocean ascension portals. So, music of the, wait. Oh, her doctorate is in music of the spheres. Wait. What did you send me? The what dictionary? Oh no. I'm not the only one that slid abruptly into the rabbit hole. Everyone has to come with me into this rabbit hole. Oh. Oh, okay. Ooh, biofields. Okay. See, I unironically love it when people use sciency or biology sounding terms for stuff that is like fake, made up. Oh, this is so long. Oh no. It's so long. Oh, yikes. Okay. I just randomly scrolled somewhere. Let me read this. False 12 strand DNA activation programs are geared towards mon- monadic reversal, reversing the fire letter sequences in the human DNA templates to create reverse sequence 11 strand activation in humans. So human DNA will assist the fallen angelic mission of getting control of Earth's planetary shields and stargates. This is Stargate. Stargate confirmed. We did it. Ion-induced by parabolic quantum state is merging with the reverse temporal reality. Science Stream, you're speaking my language. It makes I understand what you're saying. But really, Stargates. Wonderful. What if you took each letter pairings and assigned them colors and then made the image collage based on those colors? So, great. No, I know exactly what you're saying because that is what was my first thought. Going back to, publishing my DNA as some sort of like art piece or something like that, or something I could print out. Still, if you consider like 1 pixel to be like one base, well, then in standard printing format, 300 DPI, which is about 300 pixels per inch, I would still need a huge banner printed at like artistic print quality levels, which banners are not. And I think that banner had to be something like 20 by 100 feet or something like that, just to do that. Like the human genome is billions of bases long. It's huge. You're gonna buy the book of my DNA, activate your DNA with the music, ascend to an ancient, and then use your ascended abilities to clone me from the book. Sounds great. It's very pleasing that I've done all the legwork for this idea. Well, I can't help. I have to think about things while I'm eating lunch, so that's what I think about. Okay. I wanted to look at biofields, please. Biofield. Oh, wait, hold on. Oh, nope. I saw a word bioregenesis. Oh. Ooh, okay. All humans can reverse mutate DNA template distortions. Those are almost words, and bring dormant DNA template potentials into activation through self-generated DNA bioregenesis technologies, which the 12-strand DNA angelic human potential can be progressively restored and reactivated within the operational DNA. This is precisely what visiting fallen angelic legions do not want contemporary humanity to accomplish. Okay, cool. Neat. Ah, the poll closed. Yes. Thank you. I will sell my DNA as a book then. Uh, I'll let everyone know. Okay, I really just want to. You sent me this, so I. Oh, 12 strands shows up so much. I just want to know what they mean by 12 strands. No. Someone wrote 350 pages of this. I've never heard of this particular. Oh, it's a reference guide only. Uh, I have never heard of this particular fringe religion belief system. It's a little bit of Judeo-Christianity, a little bit of, because I keep seeing, I recognize one of these. Um, groups of people, like historic groups of people. I feel like I had noticed, and then a little bit of like, uh, uh, uh just biology technology terms thrown in, like black sun DNA. But they didn't show me what biofields were, unfortunately, and I don't know how to read this even. Is the thing on the left how I understand the thing on the right? Oh, no. The Triveca represents the specific mathematical program that allows biofields and planetary grids to open up pillars of frequency between here, inner Earth, and the Miyagi time cycle. Okay. People become intrigued and inspired when they read how Anne Brewer received 12 strands of DNA and reached a higher level of evolutionary potential, as described in her book, The Power of 12, A New Approach to Personal Empowerment. I mean, is her new approach to personal empowerment serious genetic editing? This actually reminds me. There was a TV show. When would this be? Like mid 2000s. There were aliens involved that were like taking over Earth. It was canceled very quickly. They knew the aliens were taking over Earth, and they figured out that the aliens had not double helixes but triple helixes. Oh, look for DNA resequencer in the glossary. No, they don't have it. I mean, they left an email address, I think. We can email this person and like let them know. Yeah, right there. Like DNA resequencers are a part of the Amenti mission to help Elohim generate biofields that promote the Budaic mind in humans, or something like that. TBH, everything that talks about a higher genetic potential is a lie to you. It should be a lie to everybody. See, it's all related to what we were talking about. Because going back to Stargate, I left her alone and got distracted. Nearty, that lady. Not that lady, that lady. Yeah, the idea of perfecting humanity through genetic engineering is silly. It's kind of like, I don't even know what to say. Like humanity, every species on aggregate is a little bit like a car with a dent or two in it. Like, of course it runs. Of course it's great. Of course it's going to get you to where you go. But there's obviously flaws. I mean, a lot of us need glasses. There are certain genetic diseases that affect certain populations more. But as science streams just pointed out, like, what is perfect, right? There's always going to be, yeah, exactly. You can't define what a perfect human being would be. You could say like maybe a human free of disease, but you are never going to be free of disease. I don't really know what we consider like aging and death, but we say aging related diseases, right? A natural part of aging, let's see, like into like past like 40s or 50s is when you start accumulating not defects, not imperfections, just things don't work as well as they used to. Your body is starting to wear out, and you've still got decades ahead of you, but you start increasing the number of problems that you have. Philosophically speaking, perfect is just good enough. Which, like, I don't know what to say, like school of thought, like I know there's like utilitarianism and that sort of thing, or like Kantianism or something like that. What would you say that falls under? Because I think thinking of perfect as being good enough is closer to a healthy way of thinking about it. I feel like just talking about like a perfect anything is human or biology related is a little tricky. But yeah, no. Also, more DNA is not the answer. In fact, like let's say, well, no, this would be tough. I'm just trying to think. If you had 12 strands of DNA, 12 identical strands of DNA, yes, but 12 strands. Okay, that's great. Now, suddenly, your cells don't know how to regulate their gene expression. Suddenly, things are being turned off and probably on, and your epigenetic landscape is getting all screwy. Let's hope, okay, at least you have an even number, so the chromosomes will segregate, hopefully properly, when it's time to divide your cell. But now, suddenly, each of your cells is trying to maintain 6 times the amount of DNA every single time. In this economy, exactly. Every cells have an economy of energy. Okay, maybe that's not so bad for humans because we can pretty easily gain calories whenever we want it. But for the majority of all of life, keeping around extra DNA is kind of a detriment. The near-T episodes of Stargate pull the eugenics lever swiftly and often. Yeah, yeah, they do. Uh, yeah. Well, that's kind of like not the subtext. It's like the text, but then the subtext is kind of like how they show it really well. In this particular thing, the bad person is the eugenicist, and the bad person literally does not create a perfect human being. She is unable, using her superior alien intellect and this god machine, she is unable to produce a perfect human, and in fact produces imperfect humans because it's complicated. You're in favor of longer quality of life. Getting rid of dementia, for instance, would be fantastic. Yeah, exactly. That's I don't know. I feel like every generation has always wished for a longer but more productive life. And I'm just curious, like, and then every generation is like, are we the ones who do it? Because it's hard to say, I'm in my early 30s. 30 years from now, are we going to have created the technology that will sustain my quality of life for a really long time? Or will it be another couple generations down the line? Yeah, I love the idea of or the implication of not just immortality, but the implications of having a healthy life that is also extremely long, right? The idea that you can be productive in like whatever sense you want that to mean, productive well into your 70s, 80s, 90s even. There are some frogs that are dodecaploid. What? Okay. I've escaped the chelonic whatever. Ooh. Although I am going to come back to that. Get rid of my optical tweezers. What species of frog is that? I'm really curious. I guess I could just do dodecaploid frog. if I'm correct, dodecaploid, dodeca, that means 12, right? So they have a ploidy of 12. Some clawed frog. Okay. Oh, here it is. The Lake Oku clawed frog. Xenopus longipus. It looks like a normal African clawed frog. That's interesting. So how are they? Why? Why though? They just published the genome like yesterday or today. Okay. Oh. Okay. Xenopus longipes genome. That's a genome I want to look at. Oh yeah, it's on bioRxiv. Is this it? So genome and cell size strongly correlated across species and influence physiological traits like developmental rate. Although size scaling features such as the nuclear cytoplasmic ratio are precisely maintained in adult tissues, it's unclear when, during embryonic development, size scaling relationships are established. Very true. Wow, redundancy rules the world. Yeah, so these, they have to have they have 12 copies of everything. I mean, it's not always true, but generally heterozygosity, which is to say a difference between the gene that you got from your mom and the gene that you got from your dad, generally a good thing. The only way to increase heterozygosity besides outcrossing, which means being crossed with individuals that are from populations that were far separated from yours, is to increase the ploidy, or the copy number of the chromosomes in your cell. So, wait, Cetipus laevis is tetraploid? I didn't know that. Okay. Wait. 2n equals 108. So it's a small frog. Okay. So is this the genome? This is just from today. Did they publish the genome of this frog? Comparison of size metrics. Okay, maybe not. Scoot down to some figures. I want to see if they have pictures of the frogs. They do. They develop much more slowly. That's interesting. I wonder if they develop more slowly because it takes longer for them to replicate their genomes. So the one thing you don't want to do when you are dividing and when you're all of life, our main job is just to sit here and divide, but especially embryos, their one job is to divide from that single cell into 2, then 4, etc. The one thing you don't want to do is rush that and divide before all of your DNA has been copied. That would be very bad. Okay, let's compare these though. So egg volume. So we have Tropicalis, laevis, and longipes. Oh, that scared me. I just saw something fly by in my periphery, but I suppose that's the point. The cell cycle is the same. Timing? I'm not reading this, so I'm getting confused. Don't say sorry. I love it. I'm the one who put that there. I put it there. You just used it. Oh, their cells are huge because they have giant nuclei. That makes sense. Okay. Oh, that was a typo before. Okay, so they have 12N equals 108. So how many actual chromosomes do they have? What's 108 divided by 12? They have 9 chromosomes and 12 copies of each of the 9 chromosomes. And laevis, so Xenopus laevis is a really, really common frog for used in laboratory research. 18, so they also have 9 chromosomes. So that's oh, thank you for the follow, Eddie Pardai. Also welcome. I don't know how long you've been here. Froggies, frogs. Okay, I don't see anything about a genome though. Ooh, yikes. So it's great that they use the raw PDF, but if each one of those dots have to be drawn individually, that's going to take forever for people to load up their images. Oh, like that. Anyways. Ooh, this is cool. So we're looking at meiosis. So, oh, you saw this paper and another one, but you can't find the other one with the actual genome sequence. Maybe they took it off bioRxiv. Uh-oh, that's never a good sign. Because bioRxiv, people leave their trash on bioRxiv very often. So if they take it off, it probably means it was very bad. Look at all that. So the purple are the chromosomes in the center of 2 cells about to divide. The green are mitotic spindles, which are like little tiny ropes that are sort of go out and latch onto the chromosomes so that the chromosomes can then be separated into 2 different cells. And so the chromosomes line up neatly in the center. But look at, wow. Just look at how much purple there is. These things are struggling to grab every single one. I wonder if, I wonder if there's any detriment to them, like like an increased risk of like aneuploidy, or an increased protection against aneuploidy. So many chromosomes. Yeah, very true. I mean, I'm kind of a fan. I'm coming around to the idea of lots of chromosomes. But let's say, okay, so let's say that you as an organism have evolved to be dodecaploid, like these guys, right? That's great. Huge cellular cost in terms of energy and nucleotide requirements. Actually, a bit of math I'm going to do. Hold on. I'm going to do a bit of math in a second. Like, what percentage of their DNA, or no, what percentage of their body mass is the extra DNA relative to Xenopus laevis, which is the same number of chromosomes but is tetraploid. And you wonder if this has to do with the survivability or fitness since they're critically endangered. Yeah, exactly. So anyways, I wonder if this does, like, one of the benefits though, I wonder, is protection against aneuploidy. So aneuploidy is when a cell divides, but instead of equally dividing the chromosomes, it's unequal. And that can be bad, especially for organisms that are diploid, because then suddenly you get a cell that potentially is missing one of the 2 parental chromosomes and is now haploid. It has only 1 copy. And that leads to other downstream problems besides just the fact that you're missing half of the DNA content of a chromosome. But if you have 12 and suddenly you have 13 in one, 11 in the other, maybe that's not a big deal. Maybe that's, they're robust to that. Okay, I got to go back up to that table though. I need to know their genome length. As all these things load. Did I already go by it? So this is something that is kind of interesting because I remember reading once someone did a calculation. So for example, people with Down syndrome, they have an extra chromosome. And I remember reading just a thought experiment of like, okay, if you took a person with Down syndrome and a person without Down syndrome, like if they were identical twins except that one had Down syndrome, they have that extra chromosome, how much heavier weight-wise would they be between the two people? Just because of the addition of that one chromosome. Apparently the difference would be about 2 grams for an average adult, which is not a lot, but I love that that's like a measurable amount, right? Okay. They already do part of the calculation for us. Okay, so for Xenopus laevis, they have an average DNA content of 6.35 picograms per nucleus. And then for Xenopus longipes, I'm not going to get that right, we have 16 picograms. So already that's really easy. So each nucleus is 16 divided by, what did I say? 6.35. So 250% more mass per, nucleus, but per cell, even though the cell itself is probably a little bit bigger. They did erythrocyte area, epithelial cell area. Okay, so their cells are bigger too, but is it by the same amount? Yes, they did nucleus area. So does this? Let's just pretend that. Okay, so what is that? So that's about 600. So laevis is 600 micrometers squared, and then so this is cell area, and then longipes is 1250. So that's about double. So it isn't 1-to-1 in terms of cellular size. So they're a little bit more compact. But that's cool. I don't know why we're talking about this. I've already forgotten. Cool nonetheless. My little arm thingy is misbehaving. Okay. How did we get from Ascension Through the Music of the Universe to dodecaploid Xenopus laevis, or Xenopus longipes? No, don't say sorry. Oh, 12D. Yes, these frogs. They've heard the Ascension Through Music of the Universe. Of course. Ship commanders of the 12 race line. 12 race lines from all 12 dimensions landed on Earth in 2017. Their fleets will navigate into their 12 stargates. Back to Stargate in 2023. All 12 keys of the universe will unite in creating glorious new symphonies that will give birth to the divinely perfect 7th level man with the divine 12 DNA template. Wonderful. And there's the website. Frog hole. Yeah. Brian set us through a frog hole, but that's okay. If you use these frogs in Jurassic Park, can the dinosaurs ascend? Yes. Well, there's plenty of DNA to fill in all the DNA gaps. Please, if anyone knows an archaeologist or a paleontologist, send me a message. I really want to talk about about Jurassic Park, but I feel like of all things, that is something I need to find a paleontologist to talk to me about. Princess and the Frog equals Mary Magdalene and her 12 enlightened DNA strands. Yes. What's the most ploid species we know about? That's a good question. So I have an idea. Okay, wait first, David, you have to tell me. How many cells can we have? Like, are you talking multicellular? Talking animal, plant, any eukaryote? You have to tell me. It's the law. This conversation derailments are where some of the coolest and weirdest stuff happens, so you have no complaints. Oh, okay. So also conversely, that means anything I have planned is just not that fun. I'm just kidding. Oh. Anything that's alive. Okay. All right. Got it. So I have some ideas about that. I want to show you two things before I just Google search it. So ploidy is tricky. Come for the science, end up with the enlightened frogs. Sorry. Yeah. Okay. Ploidy. I'm going to admit that if you had me define exactly what ploidy meant and when ploidy applied, I don't think I can tell. I don't think I know. Ploidy is really easy when you're talking about a multicellular organism where each cell, or mostly each cell, has a single nucleus, right? But there are organisms that have more nuclei than you'd expect. So, for example, stentor. Stentor, which I think means trumpet. These guys are fantastic. The trumpeter of the microscopic symphony orchestra. I feel like this all feels so connected. Symphonies, music. Anyways, stentor is this wonderful organism right here. There we go. It is insane. It has one cell. We are looking at a single cell. Yet, even though it has a single cell, it is a eukaryote like us. So a eukaryote is a highly organized organism where our DNA is kept in compartments, and also we have other special compartments and that sort of thing. They have mitochondria, all that stuff. Despite the fact that it has one cell, which normally when you think of an organism with one cell, you think of something like an amoeba, perhaps. Yeah, Brian, see, this is the question I've sort of had, but I just haven't looked up until now. So I'm just using this before I answer that for myself because I think you're right. I'm going to look at some of my favorite organisms. So amoeba, they're also eukaryotes, big blobby things, right? There isn't an up, down, left, or right. Stentor, despite the fact that it is a single-celled organism, the cell we are looking at least most of it, it has a top, it has a bottom, it has a front, it has a back, it has a left, and it has a right. So it has a body plan. It also has a mouth. It's extremely, extremely robust against damage. You can cut the thing in half and you'll get 2 new stentors. Every time it eats something big, its mouth will get absorbed into its body and will grow a whole new mouth. The cool things about these is that these sort of like string of pearls that are going down the body, these are nuclei, many, many nuclei. It has macronuclei and micronuclei. So the macro ones are these big ones, and they make all of the parts of the body. It kind of is arranged almost like a spine. And I think, if I remember correctly, that's what is hypothesized to help it sort of have a body plan. So, for example, the nuclei up front know that they're up front, so they make all the genes required for the head. The nuclei down at the back know that they're in the back, so they make all the genes required for the tail. I believe it has this string of like 10,000 copies of its genome. This, by the way, is something I've really hoped I would stumble upon on the microscope. They live in freshwater. I should be able to find one if I really looked for one. And they're very large. They're millimeters in size. Thank you for changing that color. So ploidy. So I see, this is the thing. I was told it was 10,000 ploid. But Brian, you make a very good point that ploidy is probably defined as by chromosomes per nucleus, not chromosomes per cell. That's a very important distinction. Oh yeah, for any RNA fans out there, the introns are 15 or 16 nucleotides long. The average is 15. The longest is 16. So that is the like physical limit of what needs to be like bend to make a lariat to leave between two with two exons on the sides. Okay, I'm just gonna look for stentor ceruleus ploidy. Uh-oh. Ceruleus ploidy. So let's see. Oh, a paper that I've clearly opened up before. Oh, I'm sorry. How dare I assume that RNA fan equals splicing fan? See, for everyone else, this is a great insight into people who study RNA. They're very touchy. You can't touch them or their RNA. Ploidy. Oh. Wait, what? I better not get my filthy RNases all over your RNA. Fair. I feel like there's like some joke to be made about RNA scientists not having like friends because they don't want their RNases all over them or something like that. I don't know. Give me your ploidy. So the ploidy of the stentor macronucleus is proportional to the volume of the cell. Okay. Wait, log 10. Oh, the new emote got, wait, what? The new emote got approved. Oh my gosh. Oh, they're so cute. And they almost work. Hold on, I'm checking my email right now. When did I get the email that my emote was approved? Oh. Multiple emotes got approved. That one got approved at 8 o'clock today, just as I started. And actually, they approved other emotes that I think are perhaps available. Not available, because I don't have enough emote slots. Well, there you go. There's a incentive to tell your friends and enemies to join stream, because the more people I have joining, the more emotes I can have, which has become my new goal in life, is just to have the most emotes possible because they're so cute. Anyways, what's the red? Okay, so ploidy. So the red and the black, the red and the black are two different contigs, so potentially two different chromosomes, and they have unequal ploidy, even though they're in the same nuclei. And you increase the ploidy as you increase cell volume. And it is ploidy because it is per nucleus. So Brian, you're absolutely correct, but stentor does have high ploidy. So if you, how do you reverse a log 10? I should know this. That is like 10 to the power of this number, right? So at maximum, it looks like 10 to the power of 6 ploidy. So 10 to the power of 6.5 is what? Oh my gosh. So this one is 3 million ploidy for that particular macronucleus, for that particular chromosome. For a different chromosome within the same, a different chromosome within the same nucleus, though, it's more like 10 to the 5. So let's do the lower limit. 100,000 ploidy. Okay. So David, long-winded way of saying, I think that's my answer. Somewhere between, at least for this, stentor has somewhere between 100,000 and 3 million ploidy range. So there you go. I will search, though, because that potentially is not correct or not complete. What? Actually, how would I even search this? Uh-oh. My screen's frozen. Holy fuck is right. For multicellular organisms, it looks like it's 12. Thank you. What what animals can a level 3 druid turn into? I'm running a D&D one shot in 2 days. Go away. Stop showing. What is the highest ploidy? Huh. Oh, a whole paper on it. Oh no, a whole paper on a single plant. Never mind. I think that said it had something like 200 some chromosomes, maybe. Google search suggestions can tell you so much about a person. Yeah, yeah. Back from almost falling asleep with your kid. Oh, you just missed a great I don't know, sidebar of a sidebar of a sidebar of a sidebar, talking about stentor, the amazing single-celled organism that has hundreds of nuclei, where some of those nuclei are 100 to 3 million ploid. There goes your idea that multiploidy, it's polyploidy actually, means being complex and extremely evolved. Yeah, so let's break that down actually, because it's still, I would argue, still relates to the original point of the stream, which was, improvement of like a genome. Sorry, my thing is like my computer is being slow all of a sudden. Ooh, rabbit hole. That's an emote I should make. I'll forget, so someone remind me. David, complexity, that's pretty clear. Like we can define complexity, feel like pretty easily. I think in cells so when you're talking about like cells, it's usually compartmentalization. How many compartments does the cell have? Another way of saying complexity is like organization. Are there high levels of organization, or is it that at any one point is it kind of the same? The other thing is like extremely evolved. Something I want to make very like clear to everyone, and this is not your fault for not knowing. Like only biology nerds should know this, is that every organism on Earth that is alive today is just as evolved as every other organism. Like that's the definition of life, is like life started like 1.4-ish billion years ago, and it's continued since. We are more evolved than dinosaurs. That's because they died a long time ago. But we are not more evolved than chickens, and we're not more evolved than amoebas. If you start talking about viruses, that gets a little tricky because it's unclear when those start and stop being cellular life. And so they could have arisen from bits and pieces of cellular life or something like that. But I know what you mean. Like that's why this problem, and this is what science streams brought up before, is like what the idea of like perfection. I feel like people conflate the ideas of something being extremely evolved and like extremely complex and extremely and perfected. But those are just like, you have to define your terms. And when you start defining them, you realize, oh, well, maybe they don't mean anything. Not to your detriment. Let's see. What was I... Now what was I doing? Okay, time to go back through the... okay. Sorry. Tapeworms are very well adapted for what they do. Shall we say perfected? Yeah, I mean, they are they're suited for their niche, and they do really well in their niche, meaning that their fitness is very like high. So would we say that higher fitness means closer to perfection? I don't really know. And also at potentially at some time in the future, they might no longer be perfected. I think the idea of like a perfect organism often includes the idea that they're perfect everywhere all the time. I'm trying to think. There was like Fifth Element. Fifth Element. What's her face? Lilu. Lilu? Yeah, she's like a perfect being or something like that, because she has lots and lots of extra DNA, and all that extra DNA has like genetic memory and gives her all these abilities. But does that mean she's perfected? I don't really know. Yeah. The more babies you have, then the more well evolved you are. I mean, definition wise, kind of, right? But I feel like you have to put in a little bit of like survival. Like, let's say you're an insect or something like that, and you have 100 or 1,000 babies or something like that, something ridiculous, right? But only like 1 or 0.1% survive. I don't really know. But if like most of them survive, then I would say that you're in pretty good shape. I think percent survival is really important, although I'm not an evolutionary biologist, so I don't really know how that works. Now let's get into the realm of the selfish gene. Actually, in a future stream, I don't know when I'm going to have her on. I am going to have on a scientist who doesn't study this directly, but she's very interested in this. Not just the selfish gene, but the selfish chromosome. So for those who don't know, the idea of a selfish gene is a gene who has the ability to sort of show up. How do I say this? It almost like it has its own, if it had like thoughts and it had a purpose, which it doesn't. It merely is a product of evolution. It has higher fitness than other genes like around it or something like that. So it will try to get passed on to the detriment of other genes. So it turns out it's not just genes, though. Whole chromosomes do the same thing. So I believe in humans, it's been shown that certain X chromosomes can outcompete other X chromosomes for place in the egg during oogenesis. Um, uh, the in spermatogenesis, in the creation of sperm, the single uh, gamete will eventually become 4 mature sperm, so there's no competition at that stage. Whereas in oogenesis, to create the egg, 3 become helper cells, and 1 becomes the egg that gets to live on. There are certain X chromosomes that will preferentially make their way to the egg because of the selfishness that they've evolved. But not perfect, because they don't always end up there. They just end up there at a higher rate than the others. Thus, virgin is a beta, and fuck those classifications, by the way. I mean, I don't know. You also have to think about this, like, David. It gets super complicated when you start talking about like familial groups, and I know you're joking, but it gets complicated with like familial groups that have the same genes just distributed among a small population, where it's like this potentially happens in organisms that are highly societal like us, right? And happens in ants too. Only one ant is fertile, but you get hundreds or thousands that are required to keep the line going. Or in humans, where there's not everyone has children, and not everyone is attracted to a partner that allows them to have biological children. And potentially why that has evolved is because of societal groups requiring more attention to the children who are born, and therefore there would need to be a different proportion of adults versus children than in like a mouse or something like that, where it's basically like the female, I think the male helps a little, and then that's kind of it. Whew. Anyways, there was another organism I was thinking about for ploidy, but it doesn't count as ploidy. Can someone remind me? It's T magnifica. T magnifica. Tony Hale. Thiomargarita magnifica. So this is something I talked about like a couple months ago, so some people probably missed it. This was recently discovered to be the biggest bacteria ever. So that is a United States dime right there. And these filaments, which can be seen with the naked eye, those are single bacteria. It is massive. And if I can find the paper, it's extremely complex. It does kind of like push the boundaries of what we call a prokaryote. Give me your paper. Give me your paper. Oh, Miss Rizza, you're calling it quits at once. At one, not very evolved here. No, you're evolved. You're certified evolved. Where's the preprint? Is it the DOI? They have a really great picture, like a cartoon, based on some real imaging that they took. Anyways, so this kind of pushes the boundaries of what we can really call, eukaryote versus prokaryote, because it does have organization within it that is a little bit greater than what you might find than like in other bacterium, yet is far less than a eukaryote. Okay, I am clearly unevolved, because when I'm on stream, I can never find the paper when I really want it. There is a... Ooh. No? No. Okay. Your understanding is that most of the bacteria is water vacuole. Yes, it is. And that's important because, it needs more surface, it gets over the problem of surface area to volume ratio. Eukaryotes get over that by having internal endomembrane structures, right? You need endomembrane structures to perform metabolic activities. So bacteria do a lot of their metabolic pathways, catabolic pathways, using their outer membrane, because they really only have one membrane. So they get around it by basically having a not hollow but water-filled vacuole, and this is why I'm trying to find the paper, because there's a good picture of it on the inside, so that there's this very thin layer that is the actual bacterium. I guess the vacuole is actually part of it, but there's a very thin layer on the inside going down the entire length of the bacteria. Yeah, you wonder if there's an upper limit to the size of these kinds of bacteria. So the previous winner was a different Thiomargarita species. I think it's just that they found a bigger one. So it just keeps getting lifted up and up. Can someone else find this? Why am I unable to find this? It's because I'm getting distracted. That's why. I just should go to PubMed. And close some of these tabs. I'm not closing the 12 DNA music. Oh, you think there's a good picture on Wikipedia? Thiomargarita magnifica. Come on. Let me see it. Give me the full text. I don't have access to it. Oh no. Wait. Okay, wait. I might have pre-downloaded this for that other stream when I talked about this. Otherwise, hey Brian, are you busy? Can you save me? Vapors, aha. Nope. You're getting it now? Thank you. What's this? Nope, that's silicon-based life. That's from another thing. Okay. I'm going to catch up on chat quick. Yeah, so vacuole, upper limit. Oh, yeah, good picture on Wikipedia. I'll look that up. I've looked it up before. Oh. Not so much. Oh, it's in our Discord message already. Haha. Thank you. Let's see. When did you say? 7/3/20. Nope. There we go. There we go. Awesome. Okay. Back in school, we did some DNA extraction from a Kiwi. So if you can visibly extract something that's not supposed to be seen, who knows what else is possible? Yeah, I mean, oh, come on. Yeah, no. So these things are extremely small. DNA is extremely small. But if you get enough of it, you can actually see it. Yeah, so extracting DNA from fruit is always a ton of fun. Okay, here we go. Here, oh yeah, then they have a tardigrade. Look at them. So we've seen tardigrades on stream, right? So these are the relative sizes of them. The tardigrades that I have often been quite small, actually. So I'm pretty, and you can see them with the naked eye. It's just that you need a good light source. So you can see them, and I have seen them. So I'm confident we'd be able to see these guys with the naked eye too. Yeah, so here we go. So like science streams, which said, the majority of this is vacuole. So that's what this big inner space is. A vacuole is just a enclosed space that usually holds like starch if you're a plant, or in this case, water. Yeah, and then plants also have central vacuoles that I think are filled with water or something like that too. It's a storage place. But the majority of the cells, or of the cells-like stuff, is on the periphery in this little edge. And so really this distance right here is about 2 micrometers. And then there's some other things. So these S's are sulfur granules. And then in here somewhere, they have, so these like bright lit up spots. Let me find, oh, here. These little dots, which I think correspond to these unlabeled little dots, are what they call pepins, I think, which means little seeds. It's like, I think the author said something about like that's what the little seeds on like strawberries are called or something like that. Pepin, I think that's right. But anyways, these are not nuclei, because this is related back to David's original question about ploidy. These are not necessarily nuclei, but this is where the DNA is contained. And it contains, I mean, every one of these blue ones is one of them. And this is a small section of the cells. So they contain hundreds or thousands. That doesn't really change the ploidy, potentially, but it just has a ton of them. Oh, here we go. So this is the cartoon that I was talking about. And you can see that it has a surprising amount of organization. So it uses sulfides as part of its metabolic pathway, so that makes it not unique, but a little bit different from something you'd find, growing in a pond, in your backyard, unless it's a very strange pond. It has that central vacuole, and it has this space that keeps these little compartments. It's a prokaryote. Is it a true membrane? So they didn't say that in this paper, at least not that I saw. So I don't know. Here, I'll just search membrane bound. I don't remember. I should say that. I don't remember. Membrane bound ATPases. Oh, oops. They have membrane bound organelles, says the title. Where? Sorry. I don't know if the pepins are. I can't see what it's highlighting. Yeah, sorry about that. I don't know. If I'm able to find that, I'll put it on Discord. Otherwise, wait, why don't we just share this to Discord, and then people can go look for themselves. But yeah. And membraneless organelles are all the rage these days. Also, I just noticed the word we were looking for, polyploid. Hey, see this one, Brian, this one is saying that their cells contain large numbers of genome copies, meaning that at least with bacteria, maybe it's nucleus independent because, they don't have nuclei. Polyploidy has been shown to decrease the selective pressure on genes, allowing for intracellular gene duplication reassortment divergence, leading to extreme intracellular genetic diversity. Now that's super cool. So, right, the there's strong evolutionary pressure for most of our cells to maintain diploid, meaning two copies, because there is a cost to having more, and you don't want to be rearranging your DNA. Now, that's not to say that we don't do that. In fact, red blood cells don't have DNA. Other immune cells go through DNA rearrangements in order to make some of the proteins required for your immune system. But if you start to evolve towards this ability to allow yourself for these changes in your DNA, then you can start to actually do some really cool things, right? You can start your evolution can start to play with the optimum configuration of your genome at any particular time within yourself as a single-celled organism. You just pulled out your genetics textbook, and for single-celled organisms, they describe ploidy as chromosomes per cell, even if they have multiple nuclei, which is probably why Stentor had so many. Yeah, so Stentor had many nuclei, and then within each nucleus, it was many ploidy. So that one, that's like a double whammy. But I think this one, this T magnifica, is highly polyploid because it has so many of these little pepins. But then it's hard to tell what exactly its ploidy is, because it seems like it has a high diversity of copy number of cells even within itself. Yeah. Anyways, ploidy. What you gonna do? Okay. This still cracks me up. Please don't buy this. Please don't buy this. But I am going to share this to Discord. And in fact, I'm going to finally do what I said I was going to do a long time ago. Whoops. Go away. Go away. I'm going to make a Discord channel. Is that what they're called? Called Secondary Literature, which is for stuff like this. Because it's literature, sure, but it's not primary literature. Or I don't want to put it in primary literature. I will put it right under second. Yeah, I will put it right under primary literature, though. All right, there you go. This is recorded in our Discord for the future. Please don't buy this. Wait, has there been music here the whole time? Hold on. Ooh. Okay, you probably can't hear this. Cause I don't know what the stream software is doing, but it's it's doing that thing where the sound is moving from my left ear to my right ear to my left ear, and it's like I can feel the sound move through my head. That must be my DNA becoming 12 DNA through the anointed Adonai harmonics. There's a whole Spotify channel and many resources to make you psychic by unlocking your 12 strands. Ooh, that's weird. Oh, they do shows. So you're debating whether talking about this bacteria will be exciting or confusing for your students when you talk about prokaryote versus eukaryote. I mean, that's a really... So, are you still teaching freshman, 9th grade? I don't... Because this is the thing that I've sort of that I have learned in my own education journey, which is that there's this point in the teenage years where I think a lot of people start to get very hardened in their ability to take in facts. And what I mean by that is that if you I truly believe from my experience that if you told like a 6 to 10 year old about prokaryotes, about eukaryotes, and told them about this like huge this bacterium, they just accept it as fact that it's like a spectrum. But I think as we get older, we really like to put things in nice, discrete categories, and it takes you a while of understanding the differences before you start to be able to accept the like exceptions and like the weird cases. Not that everyone can't. Like some people, obviously, they can hold all sorts of knowledge. Um, I mean, I guess, yeah. I would be super tempted to tell them about it, right? You could sort of frame it as like, okay, we're talking about eukaryotes, we're talking about prokaryotes, we're talking about the similarities, we're talking about the the differences between them. And then you could maybe like expand and say like, hey, this is what we're going to talk about. This is how we're going to, um, this is what you're going to be tested on. This is what we're going to be considering. But also life is super complex because you can even go even further than that. Like go to the fact that some viruses like Mimi virus are thought to be prokaryotic cells that have lost components required for life. Hey, Anthony Kelly, welcome. Um, yeah. Anyways, so it's like sort of this spectrum of stuff. You could almost talk about the things that link everything together, like the weird cases. But you probably just have to be super clear that like, oh, you don't have to know this, unless you want them to know it for like extra credit or something like that. I think you should. I think kids are smart. What am I saying? Kids are smart. They can do it. Brian, you've gone down the deepest of the deep rabbit holes during the stream, but you love this stuff. You find it hilarious. Well, that's great. Started talking about sci-fi. Went through some started talking about a resequencer. We stayed on the idea of DNA, so I'm super happy about that. Normally we get, well, not normally. We often get way more off topic. You're thinking of after they learn and do their comparisons to learn about endosymbiotic theory, you might give them an article from the Science News to talk about it. Ooh, make a read talk. Talk, read, talk, write about it. That's a good idea. I like that. Yes, please make them think about everything as a spectrum. This is something that scientists have a hard time thinking about sometimes. The fact that we cannot always put things into nice, discrete categories. It's super important for students to understand that even if they don't understand what is going on, or they realize that science doesn't fully understand everything, that it's often the case that things lie on a spectrum rather than in discrete categories. Because I feel like if you get too caught up in the categorization of everything, then you're later on in life when you're confronted with information that's sort of like somewhere in the middle, you're too hardened against that. Yeah, I don't like that in biology. Biology is too squishy for hard and set rules. Oh, I just noticed Teal'c has hair in this. That means this is a later season. You were teaching a group of 7-year-olds yesterday, and you were grouping things in different ways. Like these animals are our big cats, those are our sharks. One kid was grouping animals by numbers of legs. That's a great way to categorize things. That's that's wonderful. And that's a great activity too. He made a 10-leg group, and wrote 2 lions and a monkey. Who's to say that this child was wrong? That child was just clever. Also is doing like further groups of like categorizations because he's doing math to get his categories. I do enjoy that. Okay, so unfortunately, I see we have 12 people on. That's fantastic. Science Stream, you've joined us. We had Eddie Pardai, new follower. Thank you, everybody. But I do have to go. It's my first day of class tomorrow. I really need a good night of sleep because I have to get up